and welcome to Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and here is what I bring to you all from the world of medicine. Early life acquisition of antimicrobial resistance in newborn children from low and middle income countries. In a new study published in Nature Microbiology, Dr. Maria Carvalho, Dr. Christy Sands, and a network of international colleagues decided to look at the presence of antibiotic resistance genes or ARGs in the gut microbiota, the collection of microbes that are present in the human gut of mothers and their babies from 7 LMICs in Africa and South Asia. They recruited 35,040 mothers and 36,285 neonates from LMICs. From these, they collected 18,148 rectal swabs which were used to grow the bacteria present in these samples and assess the presence of clinically important ARGs in the microbiota of mothers and their babies. The authors found that a large number of samples carry genes linked to antibiotic resistance, suggesting that AMR is far more widespread in these settings than previously anticipated. For example, samples from around 1 in 5 neonates or 18.5% were positive for BLA-NDM, a gene that encodes New Delhi metallo-beta-lactamase, which is an enzyme that can destroy beta-lactam antibiotics, including the commonly used carbapenems, resulting in the bacteria being resistant against the drug. Importantly, the researchers found that ARGs were present in neonates within hours of birth, indicating that initial colonization of newborns with antibiotic-resistant bacteria occurred at birth or soon after, likely through contact with the mother or from the hospital environment. The study highlights that better understanding the routes of ARG transmission, including mother to child and within the clinical environment, is essential to prevent neonatal sepsis. Finally, the results reinforce the importance of access to safe water, sanitation, and good hygiene to reduce AMR and lower neonatal sepsis and mortality rates in low and middle income countries. Better ready-made CAR T cells for cancer immunotherapy. A technique developed by the lab of George Q. Dale, MD, PhD in the Boston Children's Hospital Stem Cell Program could make CAR T cell therapy more widely accessible. It can be difficult to gather enough functional T cells from patients' blood, and manufacturing CAR T cells for each individual patient is expensive and takes time. Time patients may not have on their side. Using induced pluripotent stem cells or iPS cells, Dale and his colleagues developed a method to make generic CAR T cells that could be produced at scale for use in multiple patients. While iPS cells are in theory a limitless source of different cell types, Dale, first author Ranjing, PhD, and their colleagues had to overcome the challenge of deriving mature, fully functioning T cells from which CAR T cells could be made. In the past, researchers have struggled with this because of the tendency for iPS cells to produce immature embryonic cells in the petri dish. Looking at epigenetic factors involved in blood development, the team zeroed in on the enzyme EZH1, which restricts the differentiation of mature lymphoid cells. Suppressing EZH1, they found, gave iPS cells the ability to make mature T cells. The team also developed a culture system that avoids co-culture with mouse-derived cells, which is cumbersome and yields T cells that aren't sufficiently mature. When the iPS cell-derived T cells were further transformed into CAR T cells, they showed anti-tumor activity comparable to that of CAR T cells derived by methods currently used for clinical therapies. These new cells had an enhanced ability to kill cancer cells in the lab and to clear cancer cells in live mice when compared to T cells made with prior iPS cell methods. Molecular map that reveals insights into the genetic drivers of chronic lymphocytic leukemia. To provide a more thorough understanding of the biological underpinnings of CLL and its molecular subtypes, scientists set out to construct a map from the largest CLL data set to date. A newly constructed map of the landscape of genetic changes in chronic lymphocytic leukemia or CLL, a type of cancer of the blood and bone marrow that exists in diverse forms and arises from various causes, provides a better understanding of this complex malignancy that could lead to more accurate prognosis for patients, improved diagnostics, and novel treatments. The work is published in Nature Genetics. To build the CLL map, the team analyzed variations in genetic sequences, gene expression patterns and chemical modifications to DNA or genomic, transcriptomic and epigenomic data from 1148 patients. The scientists identified 202 genes, 109 of which were novel that when mutated could potentially drive CLL, and they refined the characterization of subtypes of CLL with distinct genomic characteristics and prognosis. Beyond genetic sequences, the expression patterns of certain genes further subcategorized CLL and provided valuable prognostic information. Patients' clinical outcomes were associated with a combination of genomic, transcriptomic, and epigenomic features so that integrating these data could predict a patient's likelihood of experiencing remission versus developing more advanced cancer. Engineered E. coli from stool samples which can survive the hostile gut environment long enough to treat the disease. Now, a group of researchers from the University of California, San Diego successfully engineered E. coli collected from both human and mice gut microbiomes and showed that they have the potential to treat diseases such as diabetes. Their findings are published in the journal Cell. The team first collected stool samples from the host and extracted E. coli for further modifications. We say to the bacteria, hey, we'll give you a new superpower which you may not even benefit from, but we'll put you right back into the environment that you thrive in, says Zarinpur, who's the study's author. The superpower that the team gave to these specific bacteria is a protein called bile salt hydrolase or BSH. After a single treatment in mice, E. coli with BSH were found throughout the entire gut of the mice, and they retained their BSH activity for the entire lifetime of the host. The group also showed that the BSH activity was able to positively influence diabetes progression in mice. This is a significant improvement over similar treatments with non-native lab strains of engineered bacteria, where more than one treatment is often required. And these engineered bacteria do not stay in the host gut for nearly as long as or as consistently as the native E. coli method identified by Professor Zarinpar's team. In addition to successfully influencing diabetes in mice, the group was also able to make a similar modification to E. coli extracted from human gut. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.